this session, we're going to be looking at the movement of substances in and out of cells. So the three things that we will be considering would be, firstly, diffusion, secondly, osmosis, and thirdly, active transport. So first of all, when we think about movement in and out of our cell, so if we've got an animal cell here, and the kind of substances that may need to move into our cell could be things like amino acids, which are needed for nutrients. We have glucose and oxygen, which needs to move into our cells to enable respiration to occur. And then we have water, and depending on the activity of the cell, the water may either want to move in or out of our cell. And then we would have waste products being produced, so things like carbon dioxide in respiration is a waste product, and that needs to leave our cell. We could have urea, which again is a waste product and needs to leave the cell. And then we could have hormones being produced, which will be taken via the bloodstream to where they are useful. So if we consider movement of substances through diffusion, we could consider our cell again. So we have our cell and we have particles in an area of high concentration. And for diffusion, substances would move from an area of high concentration to an area of low concentration across a concentration gradient. So what that means is that because we have more particles outside the cell and less concentration of particles inside the cell, the movement of particles here would be from the outside to the inside. However, if we had a high concentration of particles inside our cell and a low concentration of particles outside our cell, then those particular substances again would move from an area of high concentration to an area of low concentration across the concentration gradient. So now let's consider the factors that affect the rate of diffusion. The four factors are temperature, distance, concentration gradient, and the surface to volume ratio. So if we look at our first factor, which is the temperature, if we increase the temperature of these molecules, then these temperatures gain kinetic energy and they will move around much faster. If they move around much faster, they are going to diffuse from the area of high concentration to lower concentration much more quickly. Now, the second thing that we were going to consider was the distance. Now, it makes sense that the further a molecule has to diffuse, the longer it takes. The shorter the distance, the shorter time taken for diffusion to occur. For oxygen molecules and the glucose molecules that are in the bloodstream outside of the alveoli, they have a very short distance to diffuse into the alveoli. Then we have the carbon dioxide, which is produced during respiration inside, and again, because the distance is very small, short distance to diffuse to outside into the bloodstream. The next factor we were going to consider was the concentration gradient. So if I have two boxes here in the same environment, if this one is box A and this one is box B. So with box A, if I have a higher concentration than in box B, but they're both more concentrated than their surrounding area, these molecules in both of them will diffuse from inside of the box across a permeable membrane to outside the box. However, A will diffuse much faster than B because the difference in the concentration gradient of A is much more different to the difference in B. So the bigger the difference in the concentration gradients, the faster the diffusion. And the last factor for us to consider would be the surface area to volume ratio. Now if we look here on the cell, we have quite a small surface area to volume ratio. So if I wanted to make that larger, I could do it in this way by increasing the surface area. And if we look here, we now have an increased surface area, which means there's more area for substances to diffuse into and out of the cell now. In general, if we think about organisms, the larger the organism, the surface area to volume ratio would be smaller. So for example, if you think about an elephant has a very small surface area compared to its actual body size, which means diffusion through the skin is very, very slow. Whereas if you have an extremely small cell, so for example something like this, the surface area compared to the volume of the cell is quite large, so diffusion happens much more rapidly. So now we've looked at diffusion and we are now going to look at osmosis. Osmosis is just like the diffusion of water. So if we're defining osmosis, we can say osmosis is the movement of water molecules across a partially permeable membrane from a region of high water concentration to a region of low water concentration. Because we do not have a high concentration of water on this side, we have a low water concentration, and I'm going to put for short for concentration, and on this side we have a high water concentration. The water molecules will diffuse through the partially permeable membrane from an area of high concentration to an area of low concentration across the concentration gradient. Now, the sugar molecules would not be able to diffuse from this side to that side because they would not fit through the spaces on the partially permeable membrane. So this is demonstrating osmosis. On the outside in the beaker, we might have a 0.7% sugar solution, and inside the whisking tubing we might have a 0.2% sugar solution. Now, because we have a 0.7% sugar solution around the whisking tubing and 0.2 inside, that means the 0.7, there's more sugar molecules in this solution, which means there's less water on the outside. Because the concentration of the sugar solution on the inside is less, that means there's more water molecules and less sugar molecules. So this means in terms of water, this would be our lower water concentration, and this would be our higher water concentration. So the diffusion of the water molecules across the partially permeable membrane, which is the visking tubing, would be from the inside, where there's a higher concentration of water, to the outside. Active transport is where substances are absorbed against the concentration gradient. So if I have my cell here and I have a high concentration of a particular substance inside the cell and a lower concentration outside, through active transport, the substances outside, the molecules outside, would be able to move against the concentration gradient to inside the cell. But this requires energy released through respiration, and that's important for active transport. You have to have energy released through respiration to enable this to happen.
Now we're going to have a look at some questions. You may wish to pause and have a go yourself before we go through the answers. So for the first question, we have a diagram which shows molecule moving in and out of the cell. The dots show the concentration of the molecules, and this cell is respiring aerobically. So if we look at question 1, it says which of those areas is going to show the movement of oxygen molecules in the cell. So if we think about oxygen molecules during respiration, oxygen moves into cells. So we're going to be looking at either A or C, but if we think of diffusion, we're looking for movement from area of high concentration to low concentration, so that has to be A. If we look at the second question, the movement of carbon dioxide, so carbon dioxide will be produced during respiration and move out of the cell, so we're either looking at B or D, but the one which is movement from area of high concentration to low concentration is B. And this process that we keep talking about of movement of molecules from area of high concentration to low concentration is called diffusion. So in this next question, we've got some students who set up an experiment to find the concentration of sucrose solution in potato cells. The student uses discs of potato cut to the same size and weighing approximately 10 grams. Then we have beakers 1, 2, 3, 4 and 5. First one has distilled water, and then we have sucrose solution of 10, 20, 30 and 40% that the discs are put into. After 2 hours, the student carefully dried the potato skins with a paper towel before reweighing the discs. So the first question is, why did the students dry the potatoes before weighing them? The answer is that you don't want the change in mass to be due to the outside water. Then for question 2, we now have the student who has recorded their results. The student recorded the initial mass, and then at the end they recorded the final mass, and depending on whether the mass of the potatoes went up or down, we have either a percentage gain or a percentage loss of mass. Now, if the final mass is larger than the starting mass, that's a gain in mass, and if the starting mass is if the starting mass is more than the final mass, then that is a loss in mass. So the first question we have is to work out the loss in mass of beaker 5. So to work out the percentage loss in mass, first of all, we have to work out what the change is. So to work out the change in mass, we can clearly see that the final mass is less than the initial mass, so we know it's going to be a loss. So to work out the change, we do the starting mass, which was 10.4 grams, take away the final mass, which was 7.3 grams, and that gives us a change of 3.1 grams. Now to work out the percentage loss, we have the change in mass, which we've just worked out as being 3 3.1, and we divide that by the starting mass, which was 10.4, and then to turn that into a percentage, we would then multiply by 100. And if we work that out, we will get our answer as being 29% loss of mass. And then for the final question, we have predict the concentration of sucrose solution in the potato cell. So we've got to use our results table here, and we can see that if we have a gain in mass, what that means is the outside solution is stronger than the sucrose solution in the potatoes. So up to beaker 2, we're having a gain in mass. And where we have a loss in mass, that means that the outer sucrose solution is less concentrated than the sucrose solution in the potatoes. So we can see that we start to have a loss in mass on beaker 3. So that means that the sucrose solution has to be somewhere in between beaker 2 and 3. And if we go back to our diagram on the other side, we can see that beaker 2 had 10% and beaker 3 had 20% sucrose solution. So that means that the concentration of sucrose solution in the potato cells has to be in between them. So you could have any answer in between 10 and 20%, and I tend to go for somewhere in the middle, so I would say 15%. But anywhere in between 10 and 20 would be correct.